Hello everyone, my name is Shashi Shrivastava and I'm from Henryford Health, Detroit, Michigan, USA. Today I'm going to talk about biomedical implants. Um, it's a physics perspective. Uh, so to start with, I would like to introduce what is the biomedical implant and why it is important in our current situation. So the biomedical implants are physical devices which can be inserted into the body to replace damaged tissues and to support specific function. So these biomedical implants are basically two types. The first type is the implants which doesn't need external support, and it is just replacing the function of a body part. For example, artificial joints, intraocular lenses, spinal implants, etc. The other type of the other category of biomedical implants which requires an external support are like power source, for example pacemakers, cochlear implants, some other biosensors, etc. So these biomedical implants are required. One of the important thing is that these biomedical implants replaces the need of medicine which is not developed yet for most of the diseases. So what is the basic challenge about these biomedical implants? Like, these biomedical implants are generally made by some external materials which are not found in the body, and these biomedical implants should be will be subjected to the physiological conditions. And I mean to say physiological conditions, I mean to say that our body is made of water and multiple ions like calcium, sodium, potassium, and so under these circumstances these devices should sustain. So the major points are like biocompatibility, size, it should be micron size or nano size, longevity, and then the bioactive properties. Some of the biomedical implants are like you need a patient specific information, like for example design, innovative structure, and there are some biomedical implants which is required to be implanted in the brain, that should be at the subcellular level. So there are several challenges, and which are like some of the examples of neural implants are like one of the device which is called cell rover, and it is used for the sensing or communication between the from the external device to the internal part of the brain, and it is developed in the MIT. And some of the subcellular devices which are used as a neural implant in brain, they require like a extensive fabrication process. The next neural implants are like the retina implants, and a retina is called as an extended brain, and it has multiple neurons, several types of neurons, and these retina get damaged because of the several diseases, for example AMD, age-related macular degeneration, and the disease like retinitis pigmentosa, which are degenerative diseases, and these retina get damaged over the time and people become completely blind. So for the cure, and there is no medicine available to cure these kind of diseases, so the only possible way is the replacing those photoreceptors available in the retina, which are responsible for the vision, with the semiconductor devices, which is called like technically biomedical implants for the retina. So some of the examples are like you can see in this picture. These are developed from the Stanford and Harvard Medical School. So these as you can see that these devices are having like several parts: external power source, goggle, camera, etc. So it's like quite cumbersome, and it is having wired structure. So we need to develop something which is like non-wired, and it can work independently inside the device, inside the body. So what we need, like we need a nano bioelectronic device which should be energy enough, having energy, like having enough power. That's why it is called energy efficient nano bioelectronic devices, and that can sustain inside the body forever. And so it becomes like a quite interdisciplinary in nature. uh, it involves material science, physical science, device physics, and bioelectronics. Apart from this, the knowledge of neuroscience is required to develop these kind of biomedical implants, which are critical for the diseases like Alzheimer, diseases like the blindness. So to give you an overview on visual prosthetics, which I said retinal implant, the AMD and RP, these diseases are like, they are the leading cause of several vision loss, and more than 30 million people in the world, they are suffering from this, and they can become partial or fully blind. So, and since due to the advancement of medical, the people are like living for a longer life, so there is more chance to get the retinal degeneration kind of diseases. So that's why it becomes a main focus area for the eye related disease. So to give you the perspective, if you see that this is the good point to start, where I can show you the picture of a brain, and you see the light is coming from the object and reaching to the retina through the optical lens, and then it is going towards the optic tract. The information is going to the optic tract, and from LGN to the optic radiations, and then it goes to the primary visual cortex, where it gives the a perfect meaning of vision. If you do the cross section of retina, you see the multiple layer in the retina, and the multiple layer, if you starting from the bottom, like inner core of the retina is having the photoreceptors, the cone and rods, which are responsible for the light absorption, and the signal is transduced to the different layers of horizontal cell, bipolar cells, amacrine cells, and then ganglion cells, and it goes to the optic nerve and to the brain for the vision processing. So if I magnify, and if you see that the cone and rods itself are very complex structure, and these rhodopsin, it is a disc-like structure. These are responsible for the transmission of like a signal. Like a signal transmission can be seen as a cystotrans changes in this rhodopsin molecules. So to give you an overall picture, the visual pathways, it starts from the eye, and then goes to the retina, back of the eye, and then goes to the brain, different part of the brain. And there are several places in the brain, A, B, C. If you see, it is marked with that. Yeah, one can have the implant, which can process the information, visual information. There are several devices developed in the late 90s and early 2000, when these are the microelectrode arrays with a limited number of arrays, and it is called Argus 1 and Argus 2. These devices becomes obsolete, but they gave like so much information for the future research, and so much idea. And based on this knowledge, what we can see that there is a development of photovoltaic retinal processes happen. And in the current scenario, like the presently, there is a FDA recognized government body. It is FDA is the government body in the US. They recognize the Prima silicon-based photovoltaic device as a like a device which can possibly replace the retina photoreceptors and give some sort of vision to the blind people. Then there is an advancement in the research, and that happened with that by the use of polymer-based semiconductors, and those are called photovoltaic retinal prosthesis. It is developed in the Switzerland, ETH, by the Dr. Gezi, and this particular retinal process is called polyretina, but it is limited by the performance of polymer semiconductors. So still, at this junction, we have these two different devices, but it's still limited to the very just for a dark and bright spot of the vision. And there are a lot of research is required to give you a device architecture basic idea. It is the semiconductor which is sandwiched between the two conductive plates, and it behaves as a capacitor, and the capacitor basic equations is applied to change the physiological condition in the eye. So I would like to introduce with some of the common biointerface materials, which are like some of them are like inorganic semiconductors like silicon based, CDSC, germano, but these are all having some other limitations because these are considered as a foreign object in the physiological medium. There are organic semiconductors which are based on the carbon and hydrogen and very close to the internal structure of the body. They can be utilized to make the organic photovoltaic devices, which can be possibly which can replace the can be used as a biomedical implant which can sustain in the body for a longer period of time. The device fabrication, this was developed by me in my lab, and I used the P3ST PCBM and ITO and germano. ITO and germano was used as an electrode, and there is another material which I use, PDB7 PC71. I tried to explore the photovoltaic performance, and I found that like the better performance of PDB7 THPC71PM. When we use this device and we use a single cell and to check the electrophysiological changes, we did the patch clamp experiment, and under the light a pulse, and we saw that when stimulation is off, when the light is off, we don't see any signal. But when we turn on the light, we see the action potential in these cells. So these kind of results were encouraging. There is another spectrum where people are doing, the scientists are doing the research, that is the nanoengineering of quantum dots, and these are the core cell quantum dots, and these are also very promising materials which can have the photoactive biointerface and which can be utilized for the retinal prosthesis. With this, I would like to conclude that the selection of the material, so the material science scope is very high in this case. We need a good device physics, design, fabrication, and tissue electrode interfacing. So these kind of it's a quite interdisciplinary work, and different multiple branch students can utilize this as a path for their future career. And at the end, I would like to show some of the glimpses from my lab, which is named as Bionics and Vision Lab. It is placed in Detroit, Michigan, and you can see it is having a dark room where we have the patch clamp and the multi-electrode array setup, and here is the retinal tissue which we did some recording, and you can see here. These are the single cell for the patch clamp recording, and it can give a very good visual glimpse how it is working in the lab. With this, I would like to thank everyone. Thank you for your attention.